Well, good day to everyone joining us and welcome to today's X Talks webinar. Today's talk is entitled AI and Machine Learning with Advanced Preclinical Cancer Models for Improved Clinical Translation. My name is Sumaya Sheikh and it's my pleasure to be your X Talks host for today. Today's webinar will run for approximately 60 minutes. This presentation includes the Q&A session with our speakers. This webinar is designed to be interactive and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll attend to your questions during the Q&A session. This chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, you can contact me at any time by sending a message using this chat panel. We also have 2 handouts available for this presentation. So audience members, you can download them through the handouts tab in your control panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Certis Oncology Solutions, who developed the content for this presentation. Certis Oncology Solutions is a life science technology company committed to accelerating the development of new cancer therapies and realizing the promise of precision oncology. Their product is oncology intelligence, highly predictive therapeutic response data derived from advanced biological models of cancer and enhanced with AI-driven bioinformatics. Certis partners with therapeutics developers to help close the problematic translation gap between preclinical studies and clinical trials. Now it's my pleasure to introduce our speakers for today's event, and our speaker is Dr. Michael Boice. Michael Boice, PhD, has over 20 years of experience in translational oncology drug development, from bench science to business development. As Senior Director of Scientific Engagement and Key Accounts at Certis Oncology Solutions, Michael works directly with biopharmaceutical clients to enhance their preclinical programs through a collaborative approach to scientific problem solving and optimized study designs. Michael's areas of scientific expertise include the development of novel oncology therapeutics, functional genomics, and rare cancers. We also have CertisAI expert panelist Dr. Long Do joining us for the Q&A session. Dr. Long Do, PhD, has over 10 years of experience advancing the application of bioinformatics and machine learning in preclinical drug development. He has helped advance many candidates through the drug discovery pipeline, overseeing bioinformatics departments supporting IND-enabling programs and successful enrollment into clinical trials. As Director of Bioinformatics and Data Science at Certis Oncology Solutions, Dr. Do leads a team of scientists responsible for the company's bioinformatics initiatives. So now, without further ado, I'd like to pass control to our speakers. So Michael, whenever you're ready, you can go ahead and get started.
Okay, great. Thank you, Sumaya. Thank you for the introduction, and thank you for everyone who joined us today. As Sumaya mentioned, today we're going to talk a little bit about some exciting things that we have going on at Certis Oncology, where we are really kind of trying to improve clinical translation using our AI, machine learning, and advanced preclinical models. So I'll jump right into the presentation here. And as Sumaya mentioned in the introduction, what do we mean by oncology intelligence? Well, at Certis, we really feel like oncology intelligence is the convergence of artificial intelligence and oncology. It's using all the information that is available, whether that's genetic information or pathological information, all the omics data that we have, using that information to define actionable results. This type of oncology intelligence has translatability across all stages of clinical drug development, has applications in clinical and translational the setting, has translational ability too in terms of oncologists, cancer patients, and drug developers as well too. It is highly predictive using our unique AI-enabled insights and validated advanced biological models as well too. So how is AI reshaping the drug discovery landscape? Well, I think this slide really kind of gives a good overview of that. As we're probably all aware, bringing a drug to market is an extremely expensive process, with estimates now being close to almost $3 billion to bring a new drug to market. Not only is it capital intensive, it's also time intensive. It takes anywhere of up to 10 years to bring a new drug to market. And the failure rate is extremely high as well too, with about a 90% failure rate. And so this is really a challenging proposition that hasn't really had much change in the last few years. But with the increase in AI capabilities and AI functionality, we see that the AI potential to reduce drug development costs can be up to 70%, not only increasing the capability of positive responses, but also removing negative responses as well too, before moving forward with costly clinical trials or preclinical development as well too. So AI is really helping to shape the drug development cost by decreasing the amount of money that's been spent. And Big Pharma has taken notice of this as well too. Over the last few years or so, their increase in R&D spending related to AI initiatives has increased by almost 22%. And this trend is predicted to continue to increase as well too. So this is a fad that is not going away, and I think will continue to shape the drug discovery landscape as we move forward. All right, so moving on to another kind of AI-directed study that was reported by McKinsey and Company last year, and I think this is a really strong study that really talks a little bit about, and this graph here really kind of shows a little bit about, how many AI drug discovery-based companies have been started in the last 10 years or so. And as you can see, there's been a steady increase in the black bars over the last 10 years or so, up until we had the last financial downturn in the early 2020s. But a continued steady increase in the number of companies that have been founded. In addition to that, these blue dots represent the number of pharma partnerships that have taken place with these AI-driven drug discovery companies as well too. And as you can see, those have increased steadily over the past number of years as well too. So really a lot of interest in terms of companies been founded, but also in terms of pharma partnerships with established biopharma companies as well too. And this slide, I think, really kind of drives it home in terms of AI-driven drug discovery companies. This talks a little bit about the funding that has been going on over the last 10 plus years or so. As you can see, a steady increase, and 2020 getting to about $6 billion or so. And a lot of this, as you can see from here, is really in late stage development as well too. So not so much pre and early stage venture, but also a lot of late stage IPO and private equity as well too. So in terms of the money is there, the companies are there, and the interest is there, I believe, from a pharmaceutical standpoint as well too. And I think that people are really starting to see how this technology might be beneficial for their drug discovery platform. So that's great. It seems to be working, or there seems to be some momentum around it, but do we have any practical applications for how it might work? And I think this is a good example of how using large genomic data sets has been beneficial in the past. So this was a publication in 2019, so a few years ago now, but it was from the AstraZeneca Sanger Drug Combination Prediction Dream Challenge. And what it was really a way to challenge individual groups or people to come up with AI-directed therapies to determine synergistic drug combinations and biomarkers with something that couldn't be done in an in vitro setting just because of the vast number of cell lines and drug combinations that they wanted to look at. So in this study, they looked at 85 or so different molecular characterized cell lines and tested almost 12,000 different drug combinations. So that in itself tells you how large this study was, and something that would be very cost prohibitive if you're doing this in a wet lab setting, but something that was able to be done in an AI setting using these large genomic characterized data sets and cell lines as well too. And the results of this were actually really quite promising, with synergy predictions being very, very accurate, over 80% in combinations, And one of them also came out with a unique synergy compound that would be beneficial in terms of targeting 2 different pathways as well too. So this is, I think, an example that shows the benefit of large genomic data sets, AI-driven therapies, and also how we might use this platform going forward. And so that's really what we're doing at Certis as well too, is we've taken large publicly available data sets with over 1 million response endpoints. And these endpoints range from a number of different points all along the drug development path line, from in vitro models to large screening, through in vivo models, so PDX models, and also into clinical candidates as well too. So we've harvested all these data sets and all these endpoints. And what we've taken from them is really 2 things. So we've taken a genetic signature as well as a small molecule structure. And we've used this to create machine learning algorithms that can predict response in vitro or in vivo. And then what also is unique about Certis is that because we have an in vivo lab setting as well too, is we can use our models and our unique PDX models and other models that we have available at Certis to validate our predictions that we get and generate from our machine learning algorithms. We can then take the models the results that we get in vivo or in vitro, and use these to retrain our machine learning algorithms, and then also go back and then revalidate the predictions that we have as well too. And so the idea here is that we have this cycle that can use the information that we've garnered from these large data sets, created our learning algorithms, validate our predictions in vivo, and ultimately to use that to make AI-directed therapies that can be tested. So how does that look when we actually do this? So this is an example of our cross-validation study that we did. So a cross-validation study, how it usually works is you take about 80% of the data that you're using and train the model on it, leaving out about 20% of the data. And then when the model is done and you're ready to input the last 20%, you do that as a cross-validation. And so you can see here in the monotherapy AI model cross-validation we did, over these 8 or 10 cancer indications that we tested, we had an accuracy rate of about 90%. So really quite good, and we were quite happy with this accuracy level in terms of the monotherapy cross-validation. And we also did this using a combination in monotherapy. And as you can see here across these 5 or 6 cell lines here, or cancer indications here, we had about an 80% accuracy as well too. So, from an initial training standpoint and accuracy standpoint, we were really quite encouraged with the accuracy levels that we received, and we're ready to continue to test this in different settings. So here's an example, and I'll walk you through this a little bit. So here's an example that we did with a couple BRAF inhibitors, dabrafenib as well as vemurafenib. So looking at dabrafenib first, so it's a V600E inhibitor. And you can see here when we used our in silico predictions in terms of response, we were able to separate out those cell lines that had V600E versus those that didn't have a V600E mutation. As you'd expect, the ones that had a V600E mutation did significantly better in terms of response. So here, the area under curve, lower response being more indicative of sensitivity. So really quite good in terms of responsiveness to our predictions here in terms of these melanoma cell lines. Now, moving on to the vemurafenib example, a lot of questions that we get asked are, what if the small molecule that we're using wasn't in the training data set? Are you able to use that and accurately predict model sensitivity or molecule sensitivity as well too? So vemurafenib was what we would call a naive model here. It was not in the trained data set, and so we didn't know how it was going to perform. And so we did a similar type analysis where we predicted its accuracy across a number of different melanoma cell lines. And as you can see here, we broke them out again as V600E versus those that were not V600E mutations. And we had a decent amount of sensitivity in the V600E versus the not V600E mutations. That we were happy with the predictions, but as you can see here, they weren't nearly as good as the dabrafenib ones. So that's two reasons. Dabrafenib is a more potent inhibitor that was more recent. Vemurafenib was an older inhibitor that was established a long time ago or approved a long time ago as well too. But we were curious, whether or not our AI really stood up to the test of these naive molecules. So we went looking for a different data set to see if we could validate our vemurafenib response information as well too. So luckily enough for us, there was a data set that was available from the Broad Institute that really was the ground truth information in terms of how vemurafenib did in these various melanoma cell lines. So you can see here, the vemurafenib on the x-axis here is the area under the curve in terms of sensitivity, and the y-axis is our predicted sensitivity as well too. And there are a few outliers, but what you can see, I think, here from the curve here and the trend is that our AI was actually pretty accurate in terms of predicting a response to this naive molecule in terms of sensitivity. So taking this information, comparing it to the ground truth and the last data on the last slide, we're really quite enthusiastic and quite optimistic about the ability of our AI predictive therapy to not only detect responses in known molecules, but also in naive molecules as well too. And so what can we do with our pre-trained AI model? And so this overview really kind of walks through a lot of what we can do utilizing our CertisAI molecule, right? So, I'll walk you through this a little bit here to explain kind of what works. So, are you working with a drug? Yes. Is it a small molecule? Yes. Perfect. Can we get the structure that you're working with and apply that to our pre-trained AI model? If that's the case, yes, then the output that we can give you from this pre-trained AI model is, like we just saw, efficacy predictions in terms of response. We can do a lot about drug synergy predictions as well too, which I'll talk about in a few slides. We can also do a lot in terms of PDX selection and validation, so applying you with the appropriate models for either cell line testing in vitro or in vivo, and then also PDX selection in terms of validations as well too. And all of this information obviously feeding into an IND therapy as well too. If you don't have a small molecule or you aren't willing to give us the structure of your target, that's okay as well too. We can work with some of the information that you have as well too. In vitro data, in vivo data from PDX models, or in vivo data from orthotopic PDX models being the best one. And we can work with any set of data, whether that's IC50, viability, tumor growth inhibition, or area under the curve information as well too, and develop these customized AI models. And I'll show you an example of what customized AI models do, but these are, more customized to the specific drug that you're working with, and we can use these to really do a better job about predicting biomarkers, PDX selection validation, and also do some information on patient stratification, looking for biomarkers and PDX selection validation as well too. So really a lot that you can do with these two different AI modules that we have, the pre-trained one or the customized AI one. And as I mentioned before on the previous slide, we also can do a lot with predicting and validating drug synergies. So, this is an example of a map that we've created in terms of all the small molecules that have been run through our AI, and it just shows you kind of an example of, what interactions we see amongst the molecules that we've tested already before. And so the idea being here that you could give us your novel compound, and we could map it and suggest where we might see different synergies based off of its structure as well too. And then obviously we can go back and test these different synergy predictions and combinations in vitro, varying the different combinations of drug B and drug A, and getting a synergy score to predict whether or not you have synergy or additive effects as well too. And we can do this across a number of different synergy score calculations from Bliss and Low, as well too. So, the ability of being able to test out synergies, I think is quite important as we move into targeted therapies that will, overcome have some development of resistance in the clinic, or also would benefit from a combination strategy in terms of efficacy. So really kind of a unique way here in terms of defining synergy in the clinic. And so I'll just do a quick summary here in terms of what we are able to do with our pre-trained model. So as I mentioned before, the inputs for predictions are really the drug target, or not the drug structure, and then the genetic gene expression profile as well too, and then also synergistic drugs that we have as well too. So all this information can be fed into our pre-trained AI model. And what we're really able to do with this information is kind of illustrated over here on the right. So, we can rank cancer types that are most likely to respond to a lead candidate and allow you to understand whether or not you should be going into lung cancer, brain cancer, sarcomas, based off the genetic signatures of those indications, as well as your molecular structure. We can select optimal models for you to test, both from an in vitro standpoint as well as an in vivo standpoint. This takes away the need to do large genomic or large in vitro screenings as well too, or even in vivo screenings. So it really gives you an idea about which models would be best to test, both from a positive sense as well as in a negative sense as well too, to validate what we've seen in an in silico prediction. It gives you the ability to prioritize your candidates. Whether or not you have a large library of things that you're working at, we can give you an idea about which ones seem to be more efficacious in different indications. So it allows you to prioritize which ones you want to be moving forward with. And then, it also, like I mentioned, it allows you to elucidate the synergistic potential of combination therapies that would be beneficial based off of your drug in different indications as well too. So, in terms of preclinical drug discovery and development, it really runs the gamut in terms of things that you might be looking for that are expensive initially to propose and do, but with this different setting, it might allow you to bypass a lot of these costs and really do a better job about model predictions and synergy predictions as well too, and different cancer types using this pre-trained AI model that we have available. Now switching gears a little bit into in vivo testing and modeling as well too. And so I think this is a great slide that really showcases a couple of things that's unique about Certis. So not only the AI directed therapies that we're doing here, but also the patient side of the business that we have. So we often work with clinical patients that are in the clinic currently, and we can take some of their tissue, grow it up in mouse avatars, and use that information, use those PDX models to indicate which type of treatments should be used going forward. So this is a particular example where we got a colorectal cancer that was metastatic in a patient. It was KRAS wild type. We took the tumor and grafted it into mice, and then also sequenced the data as well too. And based off the genetic expression that we saw within this patient, we were able to use our AI-directed therapies to predict a combination that would work well for this patient, and compare that to an oncologist-directed combination treatment, as well as a monotherapy treatment. And so what you can see here from this graph, the tumor volume, we had a significant impact in terms of response to the AI combination. So, almost a tumor stasis here in terms of tumor growth. And it did much better than the patient, the monotherapy or the oncologist recommendation chemotherapy or combination therapy as well too. So I think this is a really unique example because it highlights, what we're able to do from an AI perspective, but also what we're able to do from a patient perspective as well too. And so we have other examples of doing just what I talked about there in that last slide. Taking our PDX models that we have, for example, this was a non-small cell lung cancer KRAS model with a G12V mutation. We could take the PDX model, take its genetic information that we had, put it into a mouse, and then with our AI predictions, we could come up with a couple different combinations to test in this PDX model, and then compare that to a couple standard of cares in terms of EGFR inhibitors as well too. And so, similar to the previous example, you can see here that one of the AI combination therapies, not only had a tumor had not only tumor response, but also tumor regression in terms of response to this particular PDX model. And the other AI combination did very well as well too, over a 50% decrease in terms of tumor growth here. And very similar to what you see with the EGFR inhibitors that were treated in this model. So really exciting to see that we can then go ahead and translate this information that we get into in vivo modeling. And I think it's also important to note that I mentioned in the first slide about our AI therapy is that we can use this information that we get from these in vivo models to go back and retrain our AI data set to continue to validate it and to continue to improve its accuracy. So we've done a lot of that. And continue to improve our model. And here's just one more example to show, this one has a little bit larger in terms of different therapies that were treated. This one was done orthotopically as well too, using a KRAS G12 mutant colorectal cancer model. And again, here you can see that the AI monotherapy in green really had a significant tumor inhibition and tumor regression on this particular PDX model, whereas the combination or the combination monotherapy here from AI had about a 50% response again as well too. So we're seeing quite durable and predictive responses with our AI models in this in vivo setting. And so we've gone ahead and done this in a lot of different models. In a lot of our different PDX models, from colorectal models all the way through sarcoma models. And as you can see here, we've had pretty good results in terms of predicting efficacy in these PDX models across a number of different indications. And I know this graph's a little busy, I'm going through it a little bit quickly, but here's a better kind of summary slide to show the number of different times that we've tested this using our AI-directed therapies in orthotopic PDX models. The x-axis here being the prediction tumor growth inhibition, and the y-axis being the actual tumor growth inhibition. And so, we've had a few outliers that are bound to happen, and we can continue to use those outliers to train our model. But I think you can see here with the trend that we have here, really good in terms of accuracy and responses to what we're seeing in vivo, to what we're predicting as well with our in silico models. So this is something that we're very, very excited about, and we think has extremely a lot of potential in terms of not only preclinical drug discovery, but also in terms of clinical drug discovery as well too. So I mentioned a little bit before about that we could do custom AI models as well too. So if you come to us and you have specific data in terms of cell lines, PDX models, and or even patients, we can input this expression data into our predictive AI model and do a lot of the same things that we can do in our pre-trained AI model, but in this particular instance, it'll be custom for your particular drug of interest or the drug that you're working with. And we can also do a lot more here in terms of clinical responses, and that we can identify biomarkers that would be predictive of therapeutic responses. We can select optimal models for in vivo and in vitro validation studies, uncover biological pathways, stratify patients for clinical trial participation, and then also develop biomarker combinations or companion diagnostics as well too. So, if you have data sets that you worked with in the past that you think would be helpful to help train this model, we can certainly input that information here and use that to really kind of help you identify where you should go next with your particular MOA and molecule that you're working on. And so this is quickly an example of what we can do with the custom AI model build here. So, for example, we can run it across all different ATC cell lines because we have the genomic information for all of this. As you can see here on the x-axis are different indications, and then the y-axis is the predicted area under the curve. So the lower being the more sensitive ones as well too. So if you look over here, you can see that some of the soft tissues and sarcomas for this particular molecule that we are working with for this particular prediction seem to do better. But there is a lot of variation across these other indications as well too. There are some breast ones, some lung lines as well too, and even some cervical cancers that might do well. So here I think this custom AI model is able to show you not only indication specific, but also what types of biomarkers or different patient stratifications should you be looking for based off of the genetic signature that we're able to find through this AI program as well too. And so here's an example using our Certis, all of our Certis PDX models. And similar to what we saw in the cell lines, the sarcoma ones seem to be doing very well here in terms of prediction. But you can see here each of these individual dots represents a model. And so there's one particular ovarian model that could potentially do very, very well here, a breast line, sarcoma line, and some colorectal ones as well too. So, interesting from a perspective in terms of how you pick models and indications, but also how you pick models and what you might be looking for amongst individual models as well too. So, really, from the AI perspective, the pre-trained one or the custom model one that we have can really give you a lot of information to help you guide your preclinical efficacy studies in terms of indications and also particular models as well too. I think at that point, we'll stop, take a break, and do a quick poll question. Turn it back over to Samaya.
Thank you, Michael. And yes, so audience members, that does bring us to our first poll question. And that poll question should be on your screens right now. And that question is, what is your biggest concern, if any, with using AI and machine learning to guide your preclinical research? So select one here, and your options are, I'm worried about cost, or I don't trust the accuracy of AI predictions. That could be, it would be difficult getting approval for a new technology, or I'm worried about privacy, security, IP protection, or I have no concerns. So audience members, if you do want to send in further details, further explanations, you can do that just by using the questions chat box in our control panel. So if you just want to add more information with your answer, go ahead and do so. So once again, that question is, what is your biggest concern, if any, with using AI and machine learning to guide your preclinical research? Okay, so I'll give you a couple more seconds, audience members, to vote here, and then we'll go ahead and see the results. Okay, so I see the majority have voted. We'll go ahead and see the results now. And the results are as follows. So 5% of audience members voted for, I'm worried about cost. 48% of audience members voted for, I don't trust the accuracy of AI predictions. 5% voted for, it will be difficult getting approval for new technology. 5% also voted for, I'm worried about privacy, security, IP protection. And then 38% of audience members voted for, I have no concerns. So thank you everyone for participating. Back to you, Michael.
Okay, great. Interesting results there. Thank you, Samaya. Okay, so switching gears a little bit to the second part of our talk here in terms of advanced models. So, we at Certis are really focused on using models that we think represent what you see in a patient and what you see when you move to the clinic in a patient as well too. So, we are really focusing on determining and using advanced models that we think will enhance translational success. Whereas conventional models that are out there, for example, 2D syngeneic models and/or subcutaneously or implanted models don't really have the architecture or the molecular makeup that we would see in more advanced models. And we think that this is a key indication to why there's so much failure in terms of drug discovery and responses. And so we are really focused in on using, models that are more representative of what you see in a human setting. So, focusing on 3D, orthotopic setting, humanized models as well too. And so, here's a couple examples of that. So from our database and our models that we've used, we can show in vitro and in vivo concordance using 3DX cell cultures, how they're able to recapitulate what we would see in vivo as well too. So here's a specific example where we took a PDX model that we have at Certis Oncology, made it into a 3D cell line, and then treated it with a couple standard of care here, so erlotinib and gefitinib, and so you can see here that we have a pretty good dose response in terms of IC50 for both of them. And then what we can do then, since we have these PDX models and these 3D models as well too, is we can see if this information recapitulates in vivo as well too. So here are the in vivo test results of this PDX model that these cell lines were derived off of. And as you can see here, we have about a good response in terms of both of these agents, erlotinib and gefitinib, in this particular model. So this allows us to be able to predict whether or not, from an in vitro setting, whether or not we should be moved forward with this, because of the results seem to be pretty concordant between the two different settings. So having the in vitro model allows us to use it as a surrogate before going into more expensive, more intense in vivo models as well too. So as I mentioned earlier, Certis is very big in terms of orthotopic PDX placement and model development as well too. And for a number of different reasons, I left it over here on the left, but the main one being that we really feel like it has a much higher concordance with the human tumors. It maintains the histological characterization and really recapitulates the tumor microenvironment as well too. So we have a really outstanding technical team at Certis that has gone through a lot of training and techniques to really establish and develop these different orthotopic implantation sites for many different indications. So here is just an example of some of the ones that we've done in the past, liver, colon, lung models, GBMs in the brain, even bone models as well too. And here's an example of some MRI imaging that we've done on the tumors as well too. So, for us, we really feel like orthotopic PDX models are an advancement in terms of model placement and allow for the recapitulation of the tumor microenvironment. And so we have done a lot to make sure that we are able to implant tissues or our PDX models into the site of origin and study those on treatment studies. And so, some examples that I'll use here to really kind of showcase 2 or 3 different things that we're talking about here in terms of, does site implantation really matter? And I'll use this in the setting of a couple IO case studies. So in this particular study, we used a gastric adenocarcinoma and a 67-year-old male. As you can see here, it had some prior treatment data as well too. And some other characterization in terms of the PDX model as well too, MSI and tumor mutation burden. And this is the particular model that we're talking about here in terms of PDX models. So what we did here is we implanted the PDX model in 2 different sites. So we implanted it both subcutaneously and orthotopically in the stomach. We then did some humanization with donor PBMCs. We wanted to compare survival, CD45 chimerism, compare the proliferal leukocytes as well as TIL information to see what kind of differences we would see in terms of implantation site. So what was interesting to us to begin with was that there was no difference in terms of tumor growth and survival between the 2 different sites. So either whether it was done orthotopically in the red arrows here in circles or the subcutaneously in the gray. Survival time was pretty similar, about 40 days in terms of survival of these animals. And the level of humanization was pretty similar as well too. As you can see here, orthotopic, again, in the red arrows, and subcutaneous in the gray circles here. So the level of humanization was very similar, and the level of survival was about the same. So in terms of those 2 variables, implantation site didn't really make much of a difference. Where we did start to see some differences when we were profiling some of the TIL markers as well too. So if we look in the blood, subcutaneous is again in the gray. No PBMC injected are the first ones, and then PBMC injected subcutaneous is here. So looking at a total CD45 population, a total CD3 population, and a CD4 population, and CD8 population, there wasn't much difference, or it wasn't any significant difference in terms of infiltration in the blood. But where we did start to see a big difference is in the TILs in the tumor amongst these two different sites. So, from a CD45 population, we saw a much decreased infiltration in terms of orthotopic implantation compared to subcutaneous ones. We saw a significant difference in terms of lower amounts of CD3 positive cells in orthotopic PBMCs compared to the subcutaneous ones. Differences in CD4 as well too, although not significant, a pretty vast difference in terms of CD4 infiltration. And then again, another significant difference in terms of CD8 infiltration as well too. So, from our perspective, this does start to show why we might see different results from a subcutaneous versus orthotopic implantation setting when in the context of IO. So then again, we wanted to do something similar, but this time try with treatment. So we did a similar type of experiment that I mentioned before, where we did a sub-Q and orthotopic implantation of a specific sarcoma model. We humanized it with some donor PBMCs, and then we did treatment with PD-1. Then we wanted to look at tumor growth, TIL expression, and TIL recruitment and activation. So not similar to, or very similar to what we saw before, is that the growth didn't really see much of a difference in terms of orthotopic versus subcutaneous. The tumors grew to about 1,000 millimeters cubed between 25 and 30 days, regardless of infiltration or implantation site. But again, where we did start to see some difference here is a number of T cells within the tumor being different between the subcutaneous and orthotopic ones. And then again, particularly interesting for this case, because we use PD-1 treatment, is that we saw a significant increase in terms of interferon gamma positive, CD8 positive cells having much more expression in terms of the subcutaneous implantation versus the orthotopic implantation. So how did that translate to response? So you can see here in subcutaneous, we had tumor implantation, we had about a 30% tumor growth inhibition compared to the orthotopic, where we saw very little, if any, tumor growth inhibition. And this is kind of laid out here in the next few graphs as well too, in terms of just what the TIL infiltration looks like within the tumors in terms of PD-1 expressing CD4 positive cells, total number of CD3 positive cells, and then also the interferon gamma, granzyme B positive CD8 cells, comparing isotype treated to PD-1 treated. So, not only a significance in terms of tumor growth inhibition, but also a significant difference in terms of TIL recruitment and activation within the tumor as well too. So, just an example of showing why we believe that orthotopic versus subcutaneous has quite a bit of difference in this IO setting. We've also seen similar results in terms of small molecules and/or chemotherapy treatment, that depending on where you implant the tumor, the difference in responses can be quite significant as well too. So I know we're kind of pushing on time a little bit, so I'll jump to Samaya for the second question.
Thank you, Michael. And yes, that does bring us to our second and final poll question. And that poll question should be on your screens now. And that question is, what is your biggest concern, if any, with using orthotopic PDX, that's OPDX models, in your preclinical research? And select one here again. So your options are OPDX engrafted tumors grow slower, less reliably than sub-Q, or imaging technology is required to measure tumor volume, or OPDX models can cost more than other types of mouse models, or I have no concerns. So go ahead and click on the option that suits you best, audience members. And again, if you do have any further details, any explanations, or anything else that you want to add, you can do so just by using your questions chat box. And once again, that question is, what is your biggest concern, if any, with using orthotopic PDX, OPDX models in your preclinical research? Okay, so I see that most of you have voted, so we'll go ahead and close the poll and share the results now. And the results are as follows. So 16% of audience members voted for the first option, so that's OPDX engrafted tumors grow slower, reliably than sub-Q. less reliably, sorry, and then 37% of audience members voted for option number 2, that's imaging technology is required to measure tumor volume. 21% voted for third option, that's OPDX models can cost more than other types of mouse models, and then 26% of audience members voted for I have no concerns. So thank you everyone for participating. Back to you, Michael.
Okay, great, thank you. So yeah, I think just a few more slides to wrap up before we open it up to Q&A. So just a quick overview of Certis, talking a little bit about our service offerings. You know, as I mentioned, as we talked about in this slide, in this webinar, you know, we really have a very strong bioinformatics team that can do a lot for you, depending on where you're at various stages of the preclinical drug discovery pipeline. A lot of information that we can do in terms of model selection, synergy, compound prioritization as well too. We do a lot of custom model builds, both orthotopic, obviously our preferred, but we will do subcutaneous PDX models as well too. We understand the utility of those models as well. Humanized, both from a PBMC and CD34 engrafted setting, CDX cell lines, metastatic and resistant PDX models, the ability to do radiation, both precision and whole body. And then, as I mentioned, the PDX derived spheroids that we can use, we can do those for in vitro and in vivo, ex vivo pharmacology studies, in vivo pharmacology capabilities as well to efficacy, tolerability, PK/PD studies. Touching on one of the last polling concerns that folks have there is that, you know, we understand that orthotopic implantation requires certain imaging capabilities, and so we have you know, both MRI and BLI capabilities in-house to monitor and measure orthotopic plated tumors. We have the ability to do histology as well too on tumors, you know, and a lot of other endpoint analysis assays from flow all the way down to immunoassays, and a lot that we can do with cell-free DNA as well too. One thing that we're particularly focused on at the moment is KRAS. So one of the handouts that you actually have there that I would encourage you to download is our KRAS strategy and models that we can do. So we can really kind of do exactly what we talked about here in the webinar here. We have the ability to use our AI to help you predict combinations that might be beneficial with your specific KRAS inhibitor. Then we also have a number of different KRAS mutant PDX models that we can use to test as well too. So you know, everything that comes from this talk, we really put that together into a KRAS targeting strategy. So if you have interest in that, I would suggest that you really download the handout there that's available and look at some of the information that we have available there. And then, you know, please do feel free to get in touch. Here's some information for myself and Dr. Doe and some relevant links as well too. So thank you for joining. I appreciate everyone's time today, and I did want to leave some time for questions that myself and Dr. Doe could answer. So yeah, thanks so much.
Thank you, Michael. And yes, audience members, that does bring us to a Q&A portion for today's webinar. And we've already received some questions, so let's go ahead and start off with those. So our first question is, what data sets were used to train the pre-trained model initially? So for reference, this question was asked on slide 15.
Yeah. Long, do you want to jump in on that one, since you're the one, it's your brainchild there?
Yes, absolutely. So, you know, we're currently on our third version of our pre-trained AI model, but a lot of that clinical, you know, a lot of those clinical research studies were performed on our very first version 1, I would say. That particular pre-trained AI model for many of those clinical research studies were performed using the genomics of drug sensitivity cell lines. It's over 1 million or close to, you know, that plus actually a drug combination data set from drugcombo.org. So they also provide a lot of combination endpoints as well as the drugs themselves. And also we looked at some clinical data from TCGA. That was mostly on chemotherapy drugs. So in total, we looked at close to 4,500 drugs across the different indications that were shown.
Thank you, Dr. Doe. And we have another interesting question from an audience member. And that question is, acquired drug resistance is a big problem, and finding preclinical models that are resistant to newer therapies can be a real challenge. Can AI help get around this problem?
Yeah, absolutely. So not only are we predicting sensitivity to a drug, right? But we're also predicting when a drug is not sensitive. So in that sense, we can use our pre-trained AI models or the custom AI model that we built for you to predict models that will not respond to a drug and in that fashion test resistance.
Thank you once again. And we have another great question from an audience member. And that question is, what makes CertisAI different from other AI platforms?
Yeah, so I'm just going to speak in terms of AI and the drug discovery platforms out there. So, there are different kinds of AI platforms for drug discovery, right? So, there are those that try to find novel compounds that may hit a therapeutic target. There are AI programs, platforms that try to find a new target. And then there are AI platforms like ourselves that try to predict therapeutic efficacy. So that would be the main difference in terms of the types. Now, in terms of what we do to predict therapeutic efficacy, I think one of the big differences with us is that we're actually validating it in a really relevant preclinical model using orthotopic PDX, right? So you can make all these predictions, but as one of your concerns in the polling question was, how accurate is it, right? So really, it's about the external validation, which is the validating it, outside of the training set, right? And that's precisely what we do, right? We validate it in our OPDX models, both from our R&D models as well as in real time from our clinical patients. So I would say that really sets us apart from other platforms that may they may just test it in vitro or using other in vitro models, right?
Thank you for that comprehensive answer, Dr. Doe. And we do have another fantastic question from an audience member. That question is, you showed that an AI-chosen drug combination was effective in a KRAS-muted PDX. Would you expect similar response to that drug combination in cell lines or PDX models with the same RAS mutation?
Yeah, so I think the short answer to that is not necessarily, right? So the KRAS mutation is not the biomarker for our AI predictions, right? So the model or the patient had that mutation, but our models are actually using, or our AI models are actually using biomarkers that have a larger dynamic range. So multiple, a multivariate approach where we look at multiple biomarkers to then predict efficacy. So depending on the signature of those biomarkers in another patient or another model, that's how we'll predict it. So just because I think that's what we're finding a lot in the clinics, that even a lot of the sort of inhibitors of, for example, the BRAF inhibitors, right? If you have the V600E mutation, you see outliers, right? And then even cell lines or patients that don't have the V600E mutation still respond. So these are drivers of the cancer, but don't necessarily explain the sensitivity to the drug, right? Which is why I think our AI is also very powerful, because we're looking at not just those individual biomarkers, but multiple signals that then point to sensitivity or lack of sensitivity.
Thank you again. And another question from an audience member. That question is, would you be able to generate a custom AI model for therapeutic peptides? If so, how large a panel of cell line input data is required?
Yeah, so we absolutely can using our custom AI, custom AI platform. So that would require some sort of screening with your peptide, so some kind of screening data. And then the question then would be naturally, how large do you need? And it really depends on the indication that you're going after. But if it's a broad, if it's broad, you've tested in multiple lineages of in vitro or different cancer types and PDX. What we want to do is almost look at the results of that data and see what kind of the best training data set is where you have a variation in response, right? A variation in sensitivity. So ideally you want responders, non-responders, and everything in between, right? So if you have training data that is always responding, then there's no point, right? Or the opposite, right? So really the size of it isn't necessarily, if you have a really large set but they all just perform very similarly, that's not a good training set. So you want that variability. So it really, it's kind of a cop out, but it depends, right? So we generally, what we do is we take your data and then we plot out the response, and we can kind of tell if it's got enough. Okay, so definitely we'll work with you in terms of what that data looks like.
Thank you, Dr. Doe. And we have time for just one more question. It's a great question from an audience member. That question is, how reliable is your AI ML model for predicting time to response, complete versus partial response, degree of response, and durability of responses for monotherapy and for synergistic combination therapy? Is AUC the major predictor?
Well, so we use area under curve, especially for in vitro data, because it explains, sensitivity as well as it incorporates Cmax and some of these other things. So, versus like IC50. But we don't need AUC. In fact, when we combine data sets from PDX, so in our version 3 now, we've combined, we're combining more in vitro data. We're combining, all of the PDX data that we've learned in the past, couple years, plus, others in the past. We're combining large screening PDX studies from like Novartis and some of these other institutes that have large PDX data sets, but those have different endpoints, right? So PDX, you want to use TGI. For in vitro data, you want to use AUC, and for clinical studies, they generally use, they have classes, right? Like complete response, non-response. So those are more, you don't have that sort of continuous value of response. So what we end up doing is we normalize all the data into essentially a metric that is relative to that data set, right? So you look at, instead of AUC, you convert everything, you normalize everything to a distribution, and then we can always convert it back to whatever it is that you want to predict. So if you want to predict TGI, if you're training from AUC, you create a distribution, you train it on that distribution, and then you convert it to a TGI distribution, right? So that's kind of a really high level, or maybe not high level, but very detailed way of how we do it. But we can go from AUC to TGI to the response categories in clinical patients, right? So in PDX terms, if you want to do a complete response, generally that's like anything above, for us, like I would say 100 TGI or more would be good response, right? And if you're looking at no response, it would be like a TGI of under 50. That's sort of the accepted, industry value for this field. And anything in between that would be partial response, right? So that's kind of how we are categorizing it, but the scale there, is really about learning and figuring out what that scale is for predicting that, for the patients. Well, I hope that answers the question.
Thank you. That was very comprehensive. Thank you very much. And thank you for all of those answers and for everyone for sending all the questions in. We've reached the end of the Q&A portion of this webinar, but not to worry, if you do have further questions, go ahead and quickly jot down the emails on your screen. So for Michael and then for Dr. Joe as well, if you haven't already, go ahead and jot down those emails if you have more questions or more comments. Thank you everyone for participating in today's webinar. You will be receiving a follow-up email from XTalks with access to the recorded archive for this event. A survey window will be popping up on your screen. Your participation is appreciated as it will help us improve our webinars. Additionally, there is a link to view the recording. So I'm sending that in the chat box. So audience members, you can go ahead and also take this down. So the recording will be uploaded on the web page itself. So 1 to 3 business days after the webinar. So go ahead and take that down as well. Now, please join us in giving a big thank you to our speakers today, Dr. Michael Boice and Dr. Long Do, for that fantastic webinar. We hope you found this webinar informative. Have a great day, everyone.
Thank you.
Thank you.